your CV. See if we can get this going. All righty. So to just briefly introduce ourselves, I want to let you guys know a little bit more about the TSMA. So TSMA, Thoracic Surgery Medical Student Association, was established a few years ago, and it's now a nonprofit as of 2023. It's a national organization that really aims to recruit and put on events for medical students who may or may not be interested in cardiothoracic surgery. I think it's a fantastic initiative. I went to many events as a MS3 and really found it beneficial and really loved the mission and was fortunate to be able to join this year on the education committee. Um, I will introduce our committee members here. Carter Glenn is our, I think we can see Carter. Yep. Uh, Carter Glenn's a chair of the education committee. So he's been wonderful and helps really keep us on track. Emily Larson is also a committee member and is wonderful. I saw Julie Bushwald, who is on the research committee, and she'll probably take a bigger role in this series later on. She's on the research committee and has been wonderful as well. So many different committees, many different ways to get involved. There's many events that you all can benefit from. So I encourage you to obviously continue coming to these clinical research events. But I also think that there are many more events that students can benefit from, whether or not you're interested in cardiothoracic surgery. I think it's really applicable for anything. So spread the word. So I wanted to introduce our panel tonight and for the whole series. So from the left, Emily Larson, she's a fourth year medical student at Johns Hopkins. I am Isaac. I'm a medical student at Duke. And then I don't think Alice is on today. I think she has a conflict with interviews right now, but she's a medical student at Hopkins. And I mentioned Julie, who's a medical student at UMass. Um, and so I want to talk a little bit about the four of us. We've all had a pretty good clinical and translational research experience, I believe over 100 total publications between the four of us. So we've got a little bit of experience and I think a little bit of credibility to talk about this tonight. All righty. To hit on the objectives for tonight, we're going to start off by talking about really your introduction to clinical research. How do you get involved? Why do clinical research? And then we're going to talk about the types, the data sources, and then kind of start the introduction with the data analysis, which will be the focus of sessions 2 and 3, where you can actually apply what we're teaching you, and then we can have some time for some questions. So why do clinical research? There's a ton of reasons. A lot of people don't like research. A lot of people think research can be boring. And I think when I was a medical student, or when I was really an undergraduate student, I thought the only kind of research you can do was research in a lab setting, research working with petri dishes. And for me, that didn't sound like the most exciting or fun thing. And I didn't really want to do that. When I got to medical school, I learned about clinical research and really just learning more about being able to study the patterns and populations that we're actually treating and seeing if your interventions actually work and addressing health disparities. I mean, clinical research and health services research are really a huge field that have no limits. But I think it's closer to the patient, a little bit closer than basic science research. And I think it is really fun to be able to study what you're actually doing. So advanced medical knowledge. Let's say you're interested in a field. We're all here interested in cardiothoracic surgery. So being able to be part of the discovery process and be part of the clinical research process makes you learn more about the field, learn more about what's going on, the problems, the challenges, and then also get to meet some mentors within the field. So I think that's wonderful. Two, I think you have to ultimately learn to critically think when you are doing research and being able to get into clinical research really allows you to develop those skills early on. You start to ask what problems exist in a specialty. You start to ask, how can you make it better? And really, how can you measure it better? So I think you start to ask yourself really important questions and learn to think more critically about the field. Second, and the third thing is, even if you don't want to do research, having done research allows you to read literature and understand it better. One piece of advice that I got as a first year medical student at Duke was if you don't know how to read the literature, you're not going to be a very good doctor for more than 5 years after your training. Things change all the time. I think it's really important to be able to read and understand the research, because I think there, unfortunately, is a lot of bad research that is published out there and research that's not impactful or in bad journals or flawed methodology. So I think having working understanding of clinical research methodology is really important. We talked about meeting other people in the field, but if all else you really hate clinical research and you've done it, um, or any type of research, you've gotten to at least improve your application, your CV for residency or job or whatever it is. So I think there's a benefit and you really can't go wrong. So some misconceptions about clinical research, and for some of you, this might be entirely new, but for some people may have worked on a lab. I think the one misconception that I hear all the time is clinical research is easy, basic science is hard. Basic science is more impactful, clinical research is less impactful. I think that is a wrong notion. I think clinical research, like basic science research, is easy to do poorly, but harder to do well. I think you really have to be rigorous in your methodology, be rigorous in the questions that you ask, and I think you can do really good clinical research that is closer to the patient and very impactful. Um, so I think that's what we really want to do today and the future sessions is really instill this idea that clinical research is worth your time, that it's something worth doing, and give you the tools to move forward so that you can ask important questions and actually make good use of your time. So great. Now I've sold you on clinical research. How do we get you to sign up? How do we get you to be involved? The reality is some programs or some institutions are more heavy on clinical research or research opportunities in general. But even if not, I think that the thing that you can do is you can look around or ask around for people who are doing clinical research, whether that is in the specialty of your choice or it doesn't have to be. I think the most important thing is actually getting into the opportunity, learning the methodology, and then you can ultimately start to find other mentors that you might be more interested in working with. So, asking around, asking faculty members for guidance and research opportunities. Often there's some research groups or lab teams that you can join. I think that's very helpful. There's, for example, at my institution, we have like a surgical oncology research group, a transplant research group, and I think it's really great. And I think very similar at Hopkins for Emily and Alice. You can also network at conferences, and I haven't done this personally in terms of looking for research opportunities, but I know other students have. And that's been really great. And then there's a number of research fellowships or scholarships that allow you to do some clinical or even translational based research with other people or other institutions. If you really want to do that, we can also send that out at a later time and compile a list for those who are interested.
Awesome. You can go to the next slide. And actually, in that light, our group published a paper with a compilation of all those opportunities. So that's highlighted at the end of this presentation. So please reach out if anyone ever needs access to that. We are happy to share. So yeah, thank you all for being here again. And thanks to Isaac for the introduction. We're going to be diving in a little bit more to the technical side of things now. And so to start off, we're going to talk about the different types of clinical research available. And so this is a big flow sheet, and we're going to break it down and go through it piece by piece. So our first branching point is our primary versus secondary research. And with that, we have first is primary research. And so primary research is probably what you're more thinking when you hear about clinical research. And so this is firsthand evidence analyzed by you. And so these are things like clinical trials, case report, database analyses, stuff like that. Secondary research, also valuable, is analysis of existing evidence. And so these are things like your systematic reviews and meta-analyses and the pros and cons to both. So looking a little bit closer at our secondary research, I think we often hear a lot of overlap between things like systematic reviews and meta-analyses, and the difference between all of them is not super clear. And so kind of on a broad scale, a literature review is a description of existing literature, kind of in qualitative terms usually. And so there's different types of that. So there's narrative, there's scoping, and there's systematic, which is the most rigorous of the literature reviews. And like I said, those are kind of a qualitative summary of existing literature. A quantitative summary of existing literature, which statistically combines the data of multiple studies, is a meta-analysis. And so that's a very, very powerful way to analyze multiple results. And that's kind of the peak quality of data that you can get. Now going back to our primary side of things, the first branch point there is experimental versus observational research. And this branch point is driven by whether the exposures are assigned by the investigator. And so you hear me talk a little bit about exposures and outcomes are kind of our two main talking points for these different research study designs. And so exposures are generally are independent variables. So that's the thing that we're changing. So that's the sex of the patient. That's what type of surgery they got. That's what type of care they got. That's what type of immunosuppressant they got. And so that's the thing that we're modifying or that we're evaluating whether it makes an impact if we didn't modify it in an observational study. And then the other end of it is our outcomes. And so our outcomes is whatever we decide is our outcome. It can be survival, it can be infection, it can be stroke, it can be reintubation. So whatever your outcome is, it's your dependent variable that you're looking for. And so again, the difference between an experimental and an observational study is whether or not the investigator is the one assigning the exposures. And so in an experimental study, those are things like our randomized clinical trials or non-randomized ones. And then on the observational side, those are things like case reports or cohort studies and a lot of the clinical database research that you'll be seeing. Now looking a little more closely at those experimental research types. So those are our clinical trials. And so our clinical trials can be non-randomized or randomized. And as we all know, our randomized clinical trials, our RCTs, are our gold standard for evaluating a new treatment or intervention. And in those cases, the exposures are randomly assigned to the participants. And there's a couple different variations within this. You can be blinded or unblinded. You can do kind of the intention to treat or not. And so variations even within that, but overall, randomized clinical trial is kind of our gold standard for evaluating a new intervention. On the other side, there's a non-randomized clinical control trial, which is kind of the next step down, and that's where the exposures are not randomly assigned. And so there's a few different phases in clinical trials, especially in the approval of new devices or medications. And so there's four main phases. The first being a phase 1 study. Sometimes you'll hear colloquially phase 0 study 2, which are usually like animal studies. But a phase 1 study is a safety study, and it's in a small group of healthy participants not affected by the disease or condition that they're looking to treat with the medication. And that helps us evaluate what safe dose is. And our phase 2 study is focused on safety and efficacy and really titrating that dose. And it's a small group of affected participants this time. Next is a phase 3 study, which is safety and efficacy again, evaluating both metrics of the drug. And this is your big study. These are the ones that are going in Nature in the New England Journal. And these are a large group of affected participants, and we are looking for primarily effectiveness and trying to see if this works. And then finally, after a phase 3 study, a drug or intervention can be approved. And after that, it requires continued monitoring, which is a phase 4 study. And the other kind of phase 4 component we can look at is efficacy and safety in a specific subset of patients. Now shifting over to the observational side of primary research, our first branch point here is descriptive versus analytical research. And so in descriptive research, there's no comparison group present. So these are things like case reports and case series, which are great for starting projects for medical students a lot of times. And especially when you're on your clerkship and in a clinical setting working with someone, if you have an interesting case or if you have a patient that has a really complex condition or a really unique treatment, these are great things to write up. And there's a lot of different avenues to publish those and present them at different conferences. And it's nice because there's not an extensive amount of analysis required, and can be a really nice kind of introduction to tie in clinical to research with someone. On the other side, there are analytical studies, and that is when there is a comparison group present. So this is the bulk of clinical research. And this is a little more doable than clinical trials, which require more resources. And so a lot of times these analytical observational studies are really, really helpful to answer clinical questions. And so these are things like our cohort studies and our case control studies, and we're going to really dive deep into the differences between those next. So looking here, so again, we have our exposures and our outcomes, and those are kind of our two metrics that distinguish these different types of studies. And so a cross-sectional study, that's just taking a snapshot in time and you're assessing the prevalence of both the exposures and the outcomes simultaneously. And you're able to assess the correlation between those two with that metric. Next are the closer two ones up here, probably are easier to mix up. And those are our cohort and our case control studies. And so in a cohort study, you're looking first at the exposure. So let's say our exposure is patient sex. So in that case, you would make a group of male participants and you would have a group of female participants, and you would look for the outcome, let's say stroke, after cardiac surgery. And so you would stratify first by male and female. And then you would look at the outcome of stroke, and you would follow them to see what the outcomes are in those two different groups. Conversely, in a case control study, we would first stratify by whether or not they had a stroke, and then we would look back to see what their sex was at the time or when they got surgery. And so those are kind of our two big branching points for analytical research. And then within a cohort study, you can have either prospective, where you're looking forward and analyzing events as they come, or you can look back on it and have both of those data points at once, but stratify them by their cohort originally. So that was a lot. Quick breeze through the different types of clinical research, but hopefully that can help you have a foundation in when you're designing studies and kind of think about the different statistics that are going to apply and the different approaches you can take. Next, we're going to talk a little bit about the different data sources you can use to address these different approaches and questions that you're going to have. So as you're approaching a clinical research project, you should be aiming to answer a specific question or test a specific hypothesis is kind of the underpinning of good science, like Isaac was talking about. So within clinical research, you're going to have multiple data sources available to answer these questions, and each has pros and cons. The highest strength answer usually comes from a secondary resource, like we talked about before, which compiles multiple primary sources into one via literature review or meta-analysis. And these can also be great introductory projects for a medical student, as they can typically be done without formal IRB approval or need for extensive resources. However, one resource I want to highlight with this that can be really, really helpful, especially if it's your first time doing a literature review, are librarians. They're really, really wonderful resources that can help you kind of set up those projects and make sure you're doing it with a methodologically sound approach. So moving on to some of our sources for primary research, the next on our list is databases. And we'll go in a second, we'll go through a few of the different databases that can be useful for CT and general surgery research. But overall, databases are very useful because they typically provide a very high sample size, much higher than an individual institution could. And this provides power and is especially helpful in cases where sample size is limited, like a rare disease or a rare exposure. The downside of databases is that they're often limited in granularity, which can make it difficult to assess the finer points of clinical questions. Next is institutional data, and this can be from one institution or the combination of multiple institutions. And the big benefit of this is you can have really, really high granularity to answer the specific clinical question that you're asking. And you also have kind of more information on the patient population and the practice. And so these can provide more details, like technical details or things on postoperative management or other interventions. And big con here is that you typically have to collect the data yourself, which is a very common medical student task, which can be time consuming and a bit painful, as all of us know. But as a team effort, it's typically more manageable, and it can provide some really interesting information on really granular parts of practice that can really be impactful in changing practice. And finally, there's other sources, like we've used EMS protocols or education data that are also available to analyze when answering a specific question. And some of these can be nice, as they don't typically, again, require IRB approval and are publicly available. So now to kind of look through some of the different databases that we have available, there are a bunch. These are some of the highlights for surgical and kind of CT and transplant research. So for surgical databases, we have the National Inpatient Sample, the ACS NISQIP database, which covers about, I think, 25 to 30% of the US population hospitals. And then under that, there's also the National Trauma Data Bank. And so all those are pretty low levels of missingness, good sources for data to answer surgical questions. And then the cool thing with these, as you become more adept in navigating them, is there's a lot of different links you can make between different databases. So that whether that's with an oncologic registry, a cost registry, or some sort of pharmacologic registry, those can be really, really helpful in innovating and providing answers to new questions that people haven't previously been able to answer. And the same thing applies with our transplant / heart failure / MCS databases. And so we have things like the SRTR and UNOS OPTN databases, and those are kind of your heavy hitters for transplant research. And those are really, really wonderful because they have every single patient that's gotten transplanted in the US since the 1980s. And so having that high level of reportedness and low level of missingness is really beneficial in evaluating those outcomes. So those are really, really good starting databases. And they have those both for our thoracic organs, or our heart and lungs, but they also have the equivalent level of reportedness for the abdominal organ transplants as well. And they have lots and lots of variables. Then we have things like INTERMAX, which is helpful for MCS, and ELSO, which reports on ECMO. So all these are really, really helpful in answering clinical questions related to heart surgery and lung surgery. and general surgery. Um, Finally, I just want to talk about some general strategies for database research, since I think that's becoming more and more of a prevalent kind of area that's appropriate for medical students to explore. So when you're starting out, data dictionaries are great. Making sure you can find that to kind of make your life a little bit easier and really understand the database. And so most of them are publicly available. And you can kind of go through those in depth before proposing a project to have a really, really good understanding of what exactly you're proposing and the specific variables available and the specific limitations you might have with variables that aren't available. And if you're wondering what variables you should be using when you're evaluating a specific database, and if they have them, looking at existing literature can be super, super helpful. So if you're doing a lung transplant project on whatever exposure, you can find another lung transplant paper from another group in recent times that did it on a similar exposure, and you can evaluate their tables and look at the variables they used and kind of look for ones that are effective, any ones that you might want to add to the existing ones that you have, and those can be really, really helpful in helping you design your study and find the appropriate statistics to use and really use as a reference. Next, collaborate with your friends. People have a lot of knowledge and a lot of experience, and I've been lucky to learn from some wonderful medical students and residents who have really wonderful experience with the databases. And so kind of learning from them and getting code from your friends and colleagues and peers and people are generally very willing to support you with this is really, really helpful in helping you get started, because it can be kind of daunting to take a project from start to finish without any of that. And so having both previous literature and colleagues as references can be really helpful in kind of getting that jump start. And then finally, use whatever coding language is best for you. People have really strong preferences. I use R, Alice uses Stata. We all use different things, and totally fine. Whatever you're comfortable with, whatever you can make the prettiest figures in, totally okay. And Isaac is going to talk through a little bit of the differences between all of them. And yeah, pass it on to Isaac.
Yeah, thank you. Great job, Emily. I think that summarizes the types and data sources really well. I think before we jump into the data analysis part, I really want to emphasize that doing good clinical research really starts with the question that you have. So really trying to figure out the type of clinical research question or project you're going to do, and then where you're going to get that data, I think are really, really important. But once you have that, then it's time for you to analyze the data. And it's kind of what I'm going to talk about now. So you have a few options. You can analyze your data, or you can get somebody else to do it. And I am a big proponent on trying to learn how to do your own statistics. I was fortunate to get a master's in clinical research where I learned how to do it, but I know a lot of different people um, learn just on their own. I think nowadays, I'll be honest, when I have to do some more advanced stuff, I will, and my code's not running, I'll throw it into ChatGPT and it comes out and it tells me why my code's not running and suggests new code. And I have, like Emily said, I use R, but like I will ask my friends who know how to use R a little bit better than I do for any help that they have. So ask around, see if there's any friends that are using a certain platform. If you have friends that are using SAS or Stata or whatever, and you might feel more comfortable with that, do it. I think just finding something that you can start with, so you can at least do some of your basic stats, because you can pay for a statistician, but obviously that costs money, or you can have somebody else do it, but then you're kind of at the mercy of their own time. So I really encourage you to take ownership and learn how to do your own analyses. With that being said, talk about four different platforms. This is GraphPad Prism. Some of you may be familiar with it. Um, for clinical research, I think it is less than ideal, but I think where I see it mostly is more in the basic and translational world. It's really easy to use. It's intuitive. It has a good comprehensive set of statistical tests, and really can be used for clinical research and basic science research. It's user-friendly. But I would say that it is limited in the advanced statistical methods and more so in the ability to handle large data sets. So when Emily and Alice and Julie and I will do a transplant project, those databases will typically have hundreds of thousands of patients. So I've never used Prism for actually using that level of data. I think I've had friends who use Prism for more institutional data cohort sizes, kind of like 50 to 300, 400 in that range. And I think Prism probably works just fine, makes nice figures. But it does cost money. And I don't know, I know at Duke, we have an institutional license that we can order. Some places are like that as well. Some labs have it, so you'd have to ask around. But if you want to use Prism to start out, or for some basic science stuff as well, I don't think you can really go wrong until you're actually using large number of data. So this is R. R Studio is what I use, what Emily uses. I actually did not know what R was until I was a third year medical student when I took some time to go get a master's, and that's kind of what we learned how to do over the course of 2 years. Um, Pros about R is that it's open source and it's free to use. It has so many different types of packages, and these packages are essentially different um, packages of statistical tests and analyses that other users have written, much smarter users than I. But it's really wonderful because it can allow you to really do almost any type of statistical test or modeling that you'd want to do. It's really great for handling large data sets. I don't know if there's a better source. I think Python might be. I think Python's less used in statistics, but and I'm not going to talk about that because it's beyond my scope. But some cons about R, because we talk about how great it is. It does take a long time to learn. It has a steep learning curve, but I will say with ChatGPT now, you can really learn how to code any language. It's pretty great. And it probably would have been much easier to have learned had ChatGPT had been around like 2, 3 years ago when I first started toying with it. But you do have to have coding knowledge. It's less user friendly. And I do think that you do need a decent amount of statistical knowledge because just because your code runs does not mean that you ran the right test or you're interpreting things properly. And then also R does require some regular updates and package management, which can kind of be a little bit of a pain in the butt sometimes, especially when you're trying to run a big analysis and it says that you need to update. So similar to R, you have SAS, and I've never used SAS. I know there's a lot of statisticians who do. I think the main reason I haven't is it is expensive. You actually have to pay for it, unlike R, but it is very similar. It's powerful and comprehensive, very great for large data sets. There's a lots of resources. So when I talk about these resources, I think there are a good amount of YouTube channels dedicated to R and SAS. There are great statistical forums that you can look up or ask questions on. And I recently did that, and there's just all these great, smart people who will uh, then just respond and give you solutions as to why your code's not running, or if you're kind of modeling your analysis in the wrong way. And I tend to see SAS a lot because the headquarters are actually in Cary, North Carolina, which is about 15 minutes away from Durham. So in the biotech industry, there's a lot of people who use SAS. It's just I don't know if that's just because of where I'm at in North Carolina, but it is widely used in that. But we talked about the downside of expense. It's expensive. It does require a steep learning curve. I don't know to what degree um, compared to R, but it has less flexibility for data visualization compared to R or Python, but I think it still is wonderful. And then, of course, it requires you to actually code. So unlike Prism, if you organize your data properly, you can have Prism just like select on the few um, columns and it will like do the analysis for you. I would say Prism is very much like Excel, but like Excel on steroids. SAS and R actually require you to know how to code. But I do think that if you have an interest in clinical research or doing this long term, or really need to get into a specialty that at the worst, very worst, you need publications, a lot of publications for, learning to code as a first or second year medical student can really pay dividends. Because once you learn code, you can do your own analyses, and then also people will approach you and say, "Oh, you know how to code. Can you help me with my analysis, or can you look at my code?" And then that increases authorship as well. So I think it's very useful and just a great skill to have. And the last one, and this is going to be what we focus on in this series is Stata. And Alice is the one who uses Stata and is the pro with that. But it's more, to my knowledge, user friendly, has better interface and syntax that is a little bit less intimidating compared to R. It has a comprehensive range of statistical methods. There's excellent data management and cleaning tools. It has great graphics and the ability to visualize your data. So when I talk about that, I mean figures, tables, plots. I think all of these packages do, or all of these programs do a really great job at helping you visualize your data, which is really important when you try to publish and present your findings at conferences. And then, like R and SAS, I think SATA is very, very widely used. It has great online support and resources. So on YouTube, you can learn. There's great forums as well. It does cost money. I don't know to if it's as much as SAS. I think it's a little bit less, but it is kind of the downside compared to something like R, where you can learn for free. It does have a bit of a steeper learning curve when you want to use more of the advanced features. It's not just like Prism where it's kind of plug and play, but you do have to learn how to do it. You're less limited, or you're more limited when it comes to more machine learning and simulation kind of analyses. But I think for the majority of clinical research, I think especially at the med student level, if you can just really learn how to do descriptive statistics, some basic regression and basic survival analysis, I think that gets you a good amount of the way and helps you be valuable. And then you can, of course, learn more complex survival models and regression modeling as you go on. And I think that's makes Stata really good, and I think a really good focus for this. I know some great researchers at Duke who actually just use Stata, and they're biostatisticians, or like they know how to use SAS and R, but they've just kind of just hung on to Stata for some of their analyses. So I really think that there's no problem with it. I mean, obviously Alice has published a lot with Stata, so you really can't go wrong. And so with that, that's really what we wanted to do for and talk sorry, that's really what we wanted to talk about this first session is really lay the groundwork for clinical research, really understand why you do it, how you do it, and then give you an overview of the data analysis methods. I think it would be a good idea if you had the time to potentially download a software package, whether if you have access to STATA and the finances have already been paid for, like in a research group, that's great. If you wanted to play around with R and try to learn, I think it's wonderful as well. There's some packages in there. So I think the earlier you can learn, the better. And I think Emily might want to talk a little bit more about a plug for the papers that the TSMA has done previously.
Yeah, lots of papers available from the TSMA. JTCBS does a lot of them. There's one in Annals too. Both are great. There's a few other ones as well, but for research experiences and for scholarships and mentorship opportunities, these are all really wonderful, and lots of great resources available clinically as well.
Perfect. Alrighty. I know we have a good amount of time for questions. So does anybody have any? And if you miss this stuff, we have a recording as well. I think Alvaro has a question.
Hi everyone, thank you for the meeting. I have a question. What is the best steps that you can take to start learning any of the softwares?
Sorry, you cut out. Did you ask what is the best stats? What?
To learn the data analysis softwares.
Sorry, was it like a stats course or did you
Yes.
I think the best steps to start learning a statistical software.
oh, Emily, if you want to talk about that, I can give my 2 cents.
Yeah, I think for me, the best experiences I had for that were trying to work on projects and having progressively more autonomy, just like we aim to in training, but trying to find someone that has a lot of experience with it and asking if you can work on a project with them and then doing as much as you're able to and then being able to go for them for help. But I think that firsthand experience is probably the best way to learn, at least for me.
Yeah, that's exactly it. I got great didactic training at Duke and Duke Clinical Research Institute, but the reality is like you don't learn until you start doing it. And for me, I had a small little project that like I did not know how to run a t-test and a chi-square test in R. I honestly like did not know what those tests really were until I got to med school. And I think finding someone who can give you a project and mentor you through it is really invaluable. Taking something small or in a small little data set or a small group of patients and learning how to do that is great. And I think there's plenty of YouTube videos. I think there are some resources. I think next time around, I'll make sure to include some resources that I've found helpful. But I think that's really the best way because you can learn in theory how to do all these different things. But like you get your data and your data might be a little bit messy or like not organized in the right way. And then now some of those things just like don't apply and you have to learn to figure it out on your own. I think just taking on a project, and I think honestly, I put in the shameless plug for ChatGPT all the time in terms of learning, in terms of helping you learn to code. I think it's really wonderful. So I think just exactly what Emily said, trying to find a project and a group that can help you out. And if not, then there are some online resources you can download R, and they have some mini data sets and some mini exercises that you can do as well.
Yeah, I think like Isaac said, don't take on the machine learning 5 million patient study for your first one. Find an appropriate one. And I think none of us know it perfectly, and none of us can code something start to finish without looking something up, but you become a little more efficient at your Googling or ChatGPT as you go along.
Thank you so much.
Yeah, of course. Any other questions?
This is being recorded, so we'll be able to post this and share this again for those who missed it. But I really think the meat of it will be in sessions 2 and 3 when we actually start talking about some of the data and how to actually analyze this stuff.
Awesome.
Alrighty. Well, if there's no other questions, we'll give you about 20 minutes back of your Friday night so you can hang out or do something fun. Not that stats isn't fun, but let's see. I think we got a question in the chat. Yeah. Amisha asked how, advice for coming up with good research questions. I think that can be difficult at the med student level. I think having great mentors, and I'm like probably add to this, but having great mentors is really the key to that. And then the other key is once you've been in, like, let's say for me, I've mostly just done transplant research and lung transplant research. It's my interest, and that's what my mentors do. Uh, and so when I first showed up, it was kind of like I asked my mentors what they wanted us to do and or what they thought were interesting, and then you start to read, and then you start to have your own questions. I really encourage you to seek out good mentors, read the literature, and then try to stay with consistent with a group. Don't bounce around with too many groups. And once you're in that group, then you get more responsibility, and then you know the questions that can be asked. But Emily, I don't know if you have any advice or Carter.
Yeah, mentors are awesome because they're in the OR, they know what's happening, they have the clinical questions. I think another really helpful way approach that I was advised at one point is look at other organs and see what things have been done recently. And so if there's a really, really cool project someone just did in heart transplant and it's similarly applicable in lung transplant, that's a great project to do. And I think I've seen a lot of people have success with that. We're actually working on one right now where they did like suicide as an outcome in abdominal transplant. Now we're doing it in heart and lung transplant. And so stuff like that where you can just translate it and you have your study really well laid out for you, which is always nice on your part. And there's interest in it because the other one was published. So that's another, I think, really helpful way to take that on. Mohammed, do you have a question?
Oh, yes. Well, first of all, thank you so much for the presentation. It was wonderful. Overseed mostly the primary points of research. My question concerns it's a bit aside of the subject, but have you got any idea on the availability of remote research positions? Have you got any experience with it? Have you had any friends who had remote positions?
Yeah, I think clinical research can lend itself. Oh, sorry.
Go ahead. Go ahead. Perfect.
Clinical research can lend itself really well to remote work. And I think because a lot of it's virtual and not requiring like inpatient samples or pipetting or stuff like that, it can be done remotely. I think seeking out mentors at things like meetings and emailing people, and sometimes people won't reply, but trying to look for people that have a history of mentoring people, especially people that have like remote positions, will be good. I don't personally have any experience with taking on a remote position, but that would be my thoughts on it.
Yeah, I think clinical research is really great for that reason. You don't have to be there in person. I know there have been people, not in our research group, but just like in adjacent research groups at Duke, that there was like a student, from they may not have been they were within the USA, but they were at a different state or whatever, and they reached out. I think you can always reach out to people. Worst somebody says is no, or worst they do is um, would just ignore you. But I think clinical research definitely lends itself to that.
Thank you for the answer. And I might add something. I think that you are somehow a bit closer to well, I am I'm rocking IMG, and I am applying for the match maybe in 2027. And I'm wondering if you're are you going to be present at the AATS 2025 in Seattle?
I'll be there this year. Uh,
I'll have the pleasure to meet you then.
That'll be awesome.
Thank you for the answer.
Yeah,
Yeah, I know that the TSMA has typically like booths present at all major conferences. So even if it isn't necessarily one of us that you meet, I think that staying involved with TSMA can help sort of bridge some of those connections that you're maybe looking for. I think that's also a great way to network and try and find some opportunities that maybe you can capitalize on from a remote location. So I think that's a great idea.
Yeah, that's a great point.
Exactly. Thank you very much.
Nice.
thank you for coming. Alrighty, any other questions?
Yeah, I'll drop in the chat. Give me like 20 seconds. And feel free to go if you don't need this. It's really.
I don't, is all that on the website as well? I think there's probably some also other good resources as well on the TSMA website if you guys haven't seen it already.
Yeah, for sure.
Yeah.
And then
That's what
the
this
JP.
recording oh, sorry, Emily.
You're so fine. Go ahead.
That's where this recording will end up living, whether or not that's in piecemeal, one session at a time, or all three in series once they're all completed. But session two and session three will be held about a month apart, just giving time for this event and their subsequent events to sort of disseminate um, across networks and CT surgery. And then we'll plan to, do this monthly and get everything up onto the website, which would be a great place for you to explore both this content and other great content that TSMA has published recently.
About the two papers we had up there.
Right.
Awesome. If there's no other question, thank you all for coming. Appreciate it. Hope it was helpful.
Awesome. Nice work, guys. Thank you all for coming. And again, this will be available to you all on the website soon. Enjoy your Friday night, guys.
Hey, Carter, were you recording
Yeah.
or somebody? Okay, because I think I recorded it as well, and I see that's still going. And
Yeah.
so, okay, great.
Yeah.